Bacterial classification system. Bacterial classification system is to place an organism in a particular group on the basis of common characteristics. Number 1 is binomial nomenclature system. It was given by Carolus Linnaeus, who was also referred to as father of classification. He proposed that genus and species represent an organism. Genus is always a noun, underlined or italicized, and is capitalized. Species is a descriptive term, underlined or italicized, and is not capitalized. Number 2 is hierarchical classification system. Carolus Linnaeus created a hierarchical classification system using 7 taxonomic categories. These are kingdom, phylum, class, order, family, genus, and species. These categories are based on shared physical characteristics within each group. This system is also referred to as phylogenetic classification, as it denotes an evolutionary arrangement of species. Beginning from kingdom, each successive level of classification becomes more and more specific. Number 3 is kingdom and domains. In 1750s, as per Carolus Linnaeus, organisms were considered to belong to one of two kingdoms. Those were Animalia or Plantae. Then in 1860, Ernst Haeckel proposed three-kingdom system: plant, animal, and protista. Later in 1969, Robert Whittaker proposed five-kingdom system: Monera, protista, fungi, plant, and animal. In last few years, three-domain system, that is bacteria, archaea, and eukarya, is being used, which was given by Carl Woese. Number 4 is bacterial taxonomy, which is based on morphology, that is cell shape, arrangement, and cell size; chemical composition, that is presence of teichoic acid and lipopolysaccharide based on staining properties; cultural characteristics that determine whether the organism is pathogenic or non-pathogenic; and various biochemical properties. Number 5 is nucleic acid based classification system. It was based on G plus C content, DNA homology value, RNA homology value. It was driven by the introduction of molecular genetic tools and conserved gene sequence as phylogenetic markers, that is 16S rRNA gene sequence. Number 6 is serology and chemotaxonomy. Merriam-Webster defines chemotaxonomy as a method of biological classification based on similarities and dissimilarity in the structure of certain compounds among the organisms being classified. The compounds studied most are proteins, amino acids, nucleic acids, peptides, etc. Number 7 is numerical taxonomy. It was suggested by Michael Edenenson in 18th century, also known as Edenensonian classification or phonetic system of classification. Here the taxonomic arrangement is based on quantification of the similarities and differences among the organisms. It is expressed numerically in terms of number of characters they share, relation to total number of characters examined. Currently, Berger's Manual of Systematic Bacteriology is a manual containing useful compilation of names and descriptions of bacteria. It is the most standard reference book accepted worldwide for identification of newly isolated bacterial types. Thank you for watching. Do subscribe the channel and hit the notification bell to remain updated for more such videos.